Thank you very much. We're twice delighted to be here. First, to be given the opportunity to present our data, but as well to be the first Latin American company presenting in the R meeting, and it's a great honor and as well responsibility for us. We attended the meeting last year, and during the gala, we were asked where you're from, and he said we're from Chile, and one of the participants said, "Oh, Chile, great. I have a cousin living in Colombia." So just to put this in perspective, next slide. So I took this from the CIA. Actually, it's an overlap of the Chilean and the North American map. And as you see, the northern part will start in Alberta, Canada, and then the southern part, which is the Patagonia, will be Mexico City. So it's quite a large country. We have many nice landscape, including the driest desert in the Atacama, the glacier where you can have your whiskey with a millennial ice, and of course the famous Torres del Paine in Patagonia. Now you will think that the Ministry of Economy of Tourism has paid my trip, which is not true. We are funded heavily by the Ministry of Economy, and in California you have the Silicon Valley, and what we're trying to create in Chile is the Silicon Valley. Basically, we have the university, we have a new clinic, a hospital has been opening recently, a consortium called Regenero, and the company I'm presenting today, Cell for Cells. The company has around 35 people, including PhDs, MDs, biotech, and management. I'm attending the meeting with Oscar Solar, the CEO, and Francisco Spinoff, who is the medical officer of the company. So this is our infrastructure. This is the hospital where we have our GMP facility. As well, we have different animal and sorting facility that is directly linked to us. The company is 5 years old. We've been able to raise $31 million, which is quite an achievement, I have to say, in the region. We have many preclinical trials, as well, more than 10 patent applications, supported by, of course, peer-reviewed publication. Our portfolio contains therapies, including preclinical and clinical trials, but as well we have device-based technology, including exosome, encapsulation of cells, and today I will focus on a potency test called Stellium. I'll give you a quick cursory glance on the therapies we're doing. So we start first we're interested in looking at post-birth tissue, and we started by characterization haploidentical different sources from the same tissue, maternal versus fetal, and then we showed that fetal part and precisely umbilical cord and chorion have superiority in terms of different biological function, and we focused on that product, and then that we called Cellistem. That's the commercial name we give to this product. And we were able to conduct 2 clinical trials. One was completed last year related to dilated cardiomyopathy, and the second one is ongoing. So the first one we had reached successfully the primary outcome in terms of ejection fraction and when comparing the treated versus the placebo. This is a 34 patient trial being injected intravenously, so it's a minimally invasive technology. And then the second trial that is ongoing with the result expected in December 2016 is as well using Cellistem in intra-articular injection. And Francisco is attending with us and he'll give you further information about the trial. And then the question is, are you ready to now start injecting patients in Chile? And the answer is no. And we're working directly with the government, with the Chilean FDA, giving them our input to create the typical regulatory framework where we are protecting patients, but as well impulsing the whole technology. So back to the devices. As you know, the bottleneck of the technology is product inconsistency. So probably this is the CSO locked in the difficulties, and then the CEO is trying to pull the technology to the market. And of course, there are many challenges, including the inconsistency of the clinical effect. So we've decided to focus on our devices on a potency test based on migration. The market need, well, if you compare what's available in terms of commercial assay, whether it's the scratch assay, the transwell migration, or the different devices, they have many limitations where you cannot have a gradient. They have poor resolution. It's an endpoint assay where you can get if the cell migrated or not, but not how much they were migrating, and so on. So what is the ideal migration device? in our eyes is a design that is easy to use, good assay resolution, rapid assessment. So we use the microfluidic technology where we are able to physically restrict cell migration in a uniaxial way to release a stable gradient of factors that are relevant to the pathology we want to treat. So the device is a low cost because it's a miniaturized system. It's really require very, very small volumes of material, but as well of cells being used. And then we're using the soft lithography as a technology to produce these devices. We tested if we were able to encapsulate gradients. This was a challenging technique. I will not enter into these details, but then using a fluorescent probe, we showed that we are successful, and the technology is robust enough to have a gradient within the device. It's very, as I said, simple to use. You actually load your cells. You require a very short volume, between 10 to 20 microliters, very reduced number of cells. And then you centrifuge, we have developed an adapter, and then so you centrifuge. It's very important to align the cells, and then you look for a migration. This is a video showing the migration of these cells. So I'm going to give you two different applications. One is research related, and the second one is bioprocess related. So in the first case study, we're looking at different subpopulation within our mesenchymal stem cell. In this case, we're looking at the CD56 positive and negative population. So we actually sort these cells out, and then I looked at how they are migrating through the device. And this is something you cannot do with typical transwell or scratch assay, because you end up having the same endpoint. So we actually loaded the hydrogel encapsulated VEGF and STF1 alpha and looked for the two subpopulation. And then with a simple software, it's a computer vision based software, we're able to look at differential migration profile. But not only this, we were able to get as well a migration range, which is very relevant to understand these two differences in the subpopulation. I've showed you one factor. You can actually make it more complex by adding different factors relevant to, again, to the clinical model you're using, to the injury. You can do a statistical model and then predict the therapeutical potency of your cells. So the second test is more about manufacturing. So here we have, we're comparing different biopreservant transportation media. Usually they advertise you can keep it at 40 degrees for up to 4 days. And here we're doing biopreservant A and B, which seems to work similar in terms of viability. But however, when we put these cells, the same cells, and different biopreservant on the migration device, here we're surprised to see a dramatic difference in terms of migration profile. Pushing further the investigation, now we focus on biopreservant B, who seems to work better. We're looking at 48 hours and 120 hours, and we have the same percentage of viability, which is great. You're saying, okay, you can keep these cells for up to 120 hours without affecting the viability and then go and inject them. However, when we look at the migration profile, where we're really astonished by this result, where we have at 48 hours, we're talking about the same cells and no variability here. At 4 hours, we have a nice migration profile, and 120, we have nothing. So if I have to present the whole, if I was given one slide presentation, it would be this one. So this means we should not only assess viability. If trypan blue is not enough, we have to give, and as well, the fitness potential of these cells before injecting. We have to know what we're injecting. And obviously, viability is not enough. Biological properties such as migration or maybe other tests are required before injecting our patient with these cells. So sounds great, so you have to tell me what's the validation. And you will not believe my word if I don't show you the validation of the device. And we choose to work on a simple model of acute kidney injury, where we injected the cells at different status. So this means fresh versus 24 hours in the biopreservant, 48 and 96. And here this is a very preliminary result. I got it only yesterday, so only showing you the bun here, where actually this is the fresh cells being able to reduce this as a saline. You have high bun levels. The cells at 24 are having the same effect on the fresh cells. At 48, start getting insignificant, and of course at 96, it's getting even worse. So this is again preliminary result, but pretty exciting. So okay, so Chile, back to Chile, we have the brightest sky, known for astronomy, and then when you have an alignment of the planets in astrology, it's called stellium. And that was the inspiration for the Spinoff company and the brand we want to give to this kid. This is the team, the people behind this work. Juan Pablo being the leader of the project with his team. They've been working hard. They've been able to develop more than 650 prototype. When I say prototypes, not only the kit, but as well accessory for this prototype and as well software. They are terminating the alpha testing, and they're moving to the beta testing. So what is the spectrum we have for this innovation? Well, you all know to move your technology to the market, you have to go through the infamous Valley of Death, where you have not only to bring your technology, but as well your expectation, hype, and resources to a certain level of development. In our case, we're faced to a double Valley of Death, because first we have to show that this work at the national level, and then the challenge is to bring it as well to the international level with the international regulation and so on. So definitely we're in need of accelerator, and this is why we're here in this meeting. And then this looks like a David against Goliath challenge, and this is where what the country's slogan is all about. And back in the 19th century, this is Arturo Prat, a national hero, where he was fighting a whole fleet of steel fleet with his wooden boat. Again, more recently, a couple of unknown Chilean were nominated for the Oscar, and the other person nominated were Pixar, and they were able to clinch the Oscar Prize. And then we feel very comparable to that analogy. So at the end, what are the next steps to success? Of course, to develop a world-class technology, and we're thinking to bring some of this technology, especially the stellium technology, to the U.S. with boots on the ground. As well, we're able to offer for people from the U.S. and another part of the world who are interested in coming to Latin America, a base and a platform for their technology. But I have to admit, before doing any type of business, you have to go through the wine challenge, which is about tasting many of the dozen of Chilean wine variety before doing any business. And I will be happy to be your personal bartender over there. So anyway, thank you very much for your attention.